Okay, today I'm going to be teaching the cell cycle. Okay, so there's different phases of the cell cycle. Starting out with interphase. This is a much longer phase than the end phase, which is the mitosis phase. And it accounts for about 90% of the cell cycle. There's a couple subcategories of interphase, including the G1 phase, that's referred to as the first gap, the S phase, which is the synthesis phase, G2 in the second gap, and then there's the mitotic phase, and it includes both mitosis and cytokinesis. And it's usually the shortest part of the cell cycle, although it is the majority of what we'll be focusing on today. And then cytokinesis is when the cell is divided to form the two daughter cells, which is the whole point of mitosis, is to create two daughter cells. Okay, so can anyone tell me what the stages of mitosis are? And that's not including interphase. Interphase is something separate. Okay, so the first phase is prophase. Then after prophase is metaphase, anaphase, and telophase. And then cytokinesis is kind of off by itself in a separate phase. So now we're going to go into what the different phases are and what they mean. Okay, so just to focus on interphase for a minute, as you can see from this graph, they're all kind of split up into this whole cell cycle is shows how long the cell is in each cycle. So G1, S, and G2 are all pretty equal, and then you've got this short bit of mitosis and cytokinesis, and that's when, so you can see down here, the two daughter cells are made. So during G1, the cell prepares for DNA replication by making a variety of proteins. Those proteins will be used to create the DNA. So then during the S phase, the chromosomes are replicated, so the amount of DNA has doubled. And you're going to be able to see that in another one of the images that I have. And then G2 creates proteins and microtubules necessary for mitosis. Okay, so here we have G2 of interphase. So as you can see here, all these squiggly lines in here, those are all chromosomes. And I'm just going to highlight them a little bit so you can see. So you can see all these little lines in here. I know it's kind of hard to see with the black, but those are all the chromosomes, and so they're replicating in here so that they can form the two daughter cells. So the nuclear envelope, which is this, encloses the nucleus, which contains the nucleolus, and that's that little thing right here. It's kind of hard to see. Let me see if I can get a different color up there. Okay, for some reason, so let me pick a color on there. Yeah, the nucleolus right here. And then two centrosomes. It's right here. So that was what was created for the G2 phase, the microtubules I was talking about. This is what's going to be used for mitosis, to kind of grab those chromosomes, and you'll see that in just a minute. And the centrosomes are regions in animal cells that organize the microtubules of the spindle, you'll see that in just a minute, and duplicated and uncondensed chromosomes are present in the nucleus. And that's what I'll show you here, is that these are all chromosomes, they're just uncondensed, so they're not in the usual format that you guys are probably used to seeing them in, kind of like that. Okay? So that's part of interphase, which is not technically part of mitosis. Okay, so the first part of mitosis that we have is prophase. So as you can see here, the chromosome fibers become more tightly coiled. That's what it's talking about, and that's how you can see them like this. They're all coiled together instead of uncondensed in the nucleus. Condensing into what is now considered chromosomes made of two sister chromatins joined at the centromeres. The nucleus disappears. The nucleolus disappears, so we can't see that anymore. That was that circle that was right here on the previous picture, and that is gone now. and the mitotic spindle begins to form. And so this is the spindle right here. We kind of saw that in the last slide, but it was more condensed. So now it's kind of starting to form and spread out. And it is composed of the centrosomes and the microtubules that extend from it. Okay, moving on. Does anyone remember what was after prophase? We have, well, okay, so next would normally be metaphase, and we're just going to show you prometaphase. It's another step that you guys aren't going to need to know, but I just want to review it so that you know that some textbooks will include it. So during this prometaphase, the nuclear envelope fragments, so you can see, that all that stuff that was holding it together, like this, it is now fragmented, and you can kind of see the remnants of that right here, but it is disappearing. And the chromosomes have become even more condensed, and now the two chromatids of each chromosome has a kinetochore, which is a specialized structure at the centromere. And some of the microtubules attach to the kinetochore, which moves the chromosomes back and forth. Okay? So you got these spindles, they're coming down. You can see what they're calling the kinetochore right here. I'm not sure if I'm saying that right, but that's what we're going to go with. And then you have the chromosome, which is attached to that, the two sister chromatids. Okay, continuing on. So then after that would be the metaphase. Okay, so metaphase, all the chromosomes are going to line up at the middle. And this is called the metaphase plate. Okay, so you're going to need to know that. The chromosomes are all at the metaphase plate, which is an equal distance between the two poles. So you've got, your north and south pole, essentially, and they're lining up at the middle. The chromosomes' centromeres lie at the metaphase plate. For each chromosome, the kinetochores of the sister chromatids are attached. Okay, after metaphase, does anyone remember? Okay, oh, sorry. There we go, anaphase. Okay, so now in this phase, it's the shortest stage of mitosis, which it only lasts a few minutes. Each chromatid becomes a full chromosome, or what is considered a daughter chromosome, and begin moving to opposite ends of the cell. The non-kinetochore microtubules lengthen, causing the cell to lengthen as well. So that's talking about these ones, the ones that aren't connected to the chromosomes. Okay, and so those are going to start lengthening themselves, and that helps to elongate the cell. So then you're going to end up eventually with two cells, as those start to pull apart, because you've got all this genetic information over here that is the same as what's down here. So all these are going to be the same. We're coming up with two identical cells. Okay, so then after that we have telophase. Two daughter nuclei form in the cell, and the nuclear envelope forms from the fragments of the parent cell's nuclear envelope. The chromosomes become less condensed, any remaining microtubules are depolymerized, and mitosis is now complete. Okay, so I want to make it clear that, so interphase, right up here so you guys remember, interphase, and cytokinesis are not part of mitosis, okay? So I'm just showing here, see, now we have that nuclear envelope that's starting to form again through here, and then you've got the nucleolus forming in there. Okay, so then after telophase, we have cytokinesis, and I know this picture looks the same, but you've got this, what's called the cleavage furrow here, and that is just going to eventually separate, and then you're going to have just two cells that are identical, and they're considered the daughter cells. So just try and copy this down so that you know what's happening during cytokinesis, and so then you're going to have the nuclear envelope with your nucleolus and these chromosomes. Okay? Okay, so here I want you guys to kind of test what you have learned, and so take a minute and look at these, and then I want you to try and tell me what each one is. So we're going to go to the next slide, and we're going to look. Okay, so what was A? Let's go back and look real quick. Okay, so here we've got them at the middle. Does anyone remember what the middle was? Lined up at the middle? Metaphase. Okay, back. So now we've got B. So you've got those spindles forming, coming together. You've got the chromosomes are condensed together. It doesn't look like we have a nucleolus in there like we do down here. So B is going to be one of those early stages. What is that? Prophase. Okay. Back to C. Just give you guys another second to look. Okay, so now these have all started to pull apart. Okay, and then remember we mentioned these spindles and they're lengthening. That is going to be anaphase. All right. Okay, a couple more. Okay, so for D, see that envelope starting to disappear right here? These are all connected together. So let's go back, or forward, I guess. And that's going to be prometaphase, which I'm going to put a star by that because you guys don't really need to know that. But it is good just to have it in mind. It just is not something that is required for this class. Okay, now for this one, you can tell that this is a very early stage. Those spindles are still condensed. You still have this nucleolus. All of the chromosomes are uncondensed in the nucleus. Yeah, you still have that nuclear envelope. So that one's going to be, let's screw this up. Oh, we don't want to do that. Interphase. And more specifically, what happens, does anyone remember during this phase of interphase when it's kind of growing? So that's going to be the G2 phase. Okay, and then lastly, this last one where it's starting to come undone, but it isn't two daughter cells yet, so that's still part of mitosis. So that's going to be telophase. Okay, so now that we have some notes over this, we're going to learn like kind of some easier ways to remember this stuff. Okay, so we've got interphase, and remember that's not part of mitosis, but this is just telling you, what is going on during that phase. So DNA is replicated. Cell builds up energy reserves and grows. Okay, so the key word is going to be increase. The cells increasing, it's growing, it's building up for mitosis, and so interphase, increase. Just try and keep those together. Okay, the prophase is packaging. The chromosomes shorten and thicken. Okay, they're kind of coming together, so packaging. Metaphase, chromosomes line up in the middle of the cell. That one's easy. Remember we had it all along that metaphase plate, so middle. Then anaphase, chromatids break apart at the centromere and move to opposite poles. So it's the one where it's pulling. Okay, they're going to separate from each other, and this is where you can kind of start to see that there's going to be two cells since you've got those chromosomes on both sides. Telophase, the two nuclei forming again, nuclear envelope reforms around each group of chromosomes. So two nuclei, telophase. Okay, so we've got interphase, increasing, prophase, packaging, metaphase, middle, anaphase, apart, telophase, two. Okay? Okay, and then here's just another one. Irene, pass me another tequila calmly. This is kind of funny. So you've got interphase, prophase, metaphase, anaphase, telophase, cytokinesis. I'm telling you guys, I wish I would have had this stuff when I was learning it because I always get that stuff confused. So, if you can just try and remember these, then that would really help. And this one, PMAT, that's easy to remember. Prophase, metaphase, anaphase, telophase. And then, you just have to remember packaging, middle, anaphase, and then telophase. What was anaphase? Yeah, I already forgot. What is it?